Really be. Yes, yeah. Humbling cross.
Yes. Online, there's one projector that's working, the other that's all right. We're going to cover neurology today, folks online and folks here. We're going to start with the intro to the nervous system presentation, then we're going to move into nervous system part 2. And then 7B, the autonomic nervous system. So those will be the 3 presentations we'll hit first. And then we will go from there. So our first one will be intro to the nervous system. Our second one we'll get into is nervous system part 2. And then 7B, autonomic nervous system. Those will be the ones that we will hit. I just don't believe that our projectors are working. Let's see. Yeah, neither of them look on right now. Okay.
I saw a video yesterday that said that if you divide, if you have your spinal cord and you draw a line in half, anything that's happening on the back side will affect more motor, and anything that is happening more on the front is more sensory. So if somebody says, like in the templates, like this patient has blah, 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 the dorsal group ganglia is affected, what would you most likely see in this patient? In the answers, just look for things that are
Motor related. That's a good strategy.
That's a good
Yes.
strategy.
Absolutely. All right. Our projectors are not working. And I don't know. Huh? Yeah, I think I'm gonna give them a call again. They're on, but they're not on. They say they're on here, but they're not on above. So it's not communicating. Not communicating. Okay, let's see. Yeah. Online, if you can see, once I get the PowerPoint up. And it looks like it's just going to be this projector for now, folks. So we'll use this side, and then we're going to work on getting the other one going. But all right, everyone online, are you able to see? Yeah, I know this one. Yeah. Everyone online, are you able to see the presentation? Say like a yes or a we can.
Yes.
Thank you. Okay, awesome. All right. Well, we're going to start, like I said, with intro to the nervous system. That's our first presentation we're going to go through. This is going to be a good review and kind of give us the different differentiations of the nervous system and The goals for this presentation is for you to be able to list the basic functions, explain the divisions, understand the different neuroglia and site locations. We've had a couple practice questions on different types of neuroglia. Be able to understand what a neuron is, its structural components, understand the difference between nucleus, ganglion, nerve, and tract. How do myelin sheaths affect the central and peripheral nervous system? And then classify different types of neurons. When they do a nervous system or a neurology, physiology question, they like to make you differentiate between different types of cells, different type of neurons. We've seen a couple questions, astrocytes and microglia, and actually wanting you to know their functions. So going through and making sure you understand the differences between the two is important. We'll then delve more into the CNS and the PNS, the central nervous system and the peripheral nervous system. And we'll spend a significant time today on the autonomic nervous system because there's a lot of questions that can be derived from that section. So again, the nervous system as a whole is our major controlling and communicating system of the body. The other is the endocrine system. The nervous system is rapid and specific and typically almost always causes an immediate response, which is how it can be different than the endocrine system. Cells are communicating via electrical and chemical signals. There's 3 different functions that we want to keep in mind when we talk about the nervous system as a whole, and that's sensory input, integration, and motor output. So again, sensory input is information being gathered from our sensory receptors about some type of internal or external change to the body. Then integration is the processing center that's typically done in our central nervous system, so brain and spinal cord. And then we have motor output, where our effector organs, both muscles and glands, are producing some type of response. So in general, those are the 3 areas you're thinking about when you have any nervous system question. What is the sensory input or the stimuli that I'm dealing with? What type of integration, where is that occurring? And then what's our motor output or the output that I'm expecting to happen from that integration? So the divisions in general, 2 major parts. We have our central nervous system and our peripheral nervous system. I'll refer to these often as CNS or PNS. The central nervous system is our brain and spinal cord of the dorsal body cavity. It's our integration and control center. It's going to interpret our sensory input and it's going to dictate what our motor output is. Our peripheral nervous system is going to be the portion of the nervous system that does not include the central nervous system. So everything that's not the CNS, you can include into your peripheral nervous system. This is going to be all of your nerves that extend from the brain and spinal cord, spinal nerves to and from the spinal cord, and cranial nerves to and from the brain. These are all your peripheral nervous system. And you can see here on that picture. Then the peripheral nervous system can be divided even further. And I apologize for my voice being gone. I was coaching yesterday, and so I was yelling at athletes and lost my voice. Anyways, so there's 2 different divisions of the peripheral nervous system, sensory or the afferent division and motor or the efferent division. Knowing afferent versus efferent is important. They will include those to try to confuse you. So understand that our afferent division includes our somatic sensory fibers and our visceral sensory fibers. So when we think of what is visceral, I'm going to start there. Visceral, we think of viscera, we think of organs. So our visceral sensory fibers are going to convey impulses that we get from our visceral organs to the central nervous system. Our somatic sensory fibers are everything else, skin, skeletal muscles, joints to the central nervous system. So it's a good way of separating. Our efferent division transmits impulses from our central nervous system to its effector organs, the organs that it's going to contact with and try to do some type of response. This could be muscles and glands, and there's 2 major divisions here, our somatic and our autonomic nervous system. So if you are not sure where to start with the nervous system, understanding these divisions would be a good place to start, making sure you can separate who does what areas are at play. So delving deeper into our somatic nervous system again, which is a component of our motor or efferent division, our somatic motor nerve fibers are going to conduct impulses from the central nervous system to our skeletal muscles. This is voluntary, so it's conscious control of our skeletal muscles. This is how we decide to actually move parts of our body. Versus the autonomic nervous system consists of visceral motor nerve fibers. It is not voluntary, it's autonomic nervous system. regulates our smooth muscle, cardiac muscle, glands, and it has two functional subdivisions, which we'll get into in our third presentation today, which is our sympathetic and parasympathetic, which work in opposition. So another way to look at kind of organization of the nervous system, again, we have our central nervous, oops, here, let me get my little pointer out. We have our central nervous system over here, our brain and spinal cord. It's our integrative and our control center. We then are going to have our peripheral nervous system on the other side, which includes our cranial nerves, our spinal nerves, and then the communication lines between the central nervous system and the rest of the body. This breaks into our sensory or afferent and motor or efferent division. Our sensory afferent division ends here, and it's our somatic and visceral sensory nerve fibers that send impulses from the body to the central nervous system. Versus our motor or efferent division, we see has several other divisions below. So our motor efferent division is our motor fibers that conduct impulses from the central nervous system to the effector muscles and glands. Our somatic nervous system is our voluntary motor nerve fibers that conduct impulses from the CNS to skeletal muscles, and we control this. Our autonomic nervous system is our visceral involuntary motor nerve fibers, again, going from the CNS to cardiac, smooth muscle, and glands. And the autonomic nervous system is the sympathetic and parasympathetic divisions that we often talk about, and we will spend time there. If you have this in your brain and you can kind of walk yourself through what is my inputs, what are my outputs, and which area is of control am I looking at, that's a great place to start. That can help you be able to logically walk your way through a lot of your different nervous system questions. And then we're going to get into the cells, which if you know those two areas, if you know this presentation, you'll be set up for pretty good success for the nervous system. Obviously, there'll be some more minutia questions that could come, but this is a good overview. So we are going to go from big picture down to more of a cellular level now, looking at our neuroglia, looking at more of our nervous tissue histology. And when we think about our neuroglia, we're thinking about two principal different cell types, our neuroglia or our glial cells, and our neurons or our nerve cells. Neuroglia is the small cells that surround and wrap around our delicate neurons versus our neurons are our nerve cells. Those are the excitable cells that we think about most commonly that are transmitting our signals. So there's four major neuroglia that support our central nervous system neurons. So the four neuroglia, four cell types we'll get into is astrocytes, microglia, ependymal, and oligodendrocytes. Those are our four main neuroglial cells that support our neuron cells. So our astrocytes are the most abundant and most versatile glial cell. They're going to cling to neurons. We can see here, this is our astrocyte in the middle, almost looks like a spider. And it's clinging on to our neuron cells. This is a capillary flowing through. So it's clinging on to our neuron cells over here. It's also going to contact synaptic endings and capillaries. So it's a good big communicator. And really what it's doing is it's providing some structural support. So it's contacting all these things with big different wings, and it's providing some structural support to brace neurons. It also plays a role in exchanges between blood and nervous tissue. It's going to help guide migrations because it's contacting the neurons, so it can help pull them or move them to certain locations. Because it can contact capillaries and blood flow, it can control the chemical environment around neurons. It can therefore respond to nerve impulses and neurotransmitters, influencing neuronal functioning by being able to communicate with the blood and the nervous system cells, and help with information processing in the brain. So when I think of astrocytes, I think of two things. I think of structural support and communication between the blood and the neurons. Those are my two big things with astrocytes. Yeah. Yes, it is. It's one of your major ways that we're actually communicating between that blood and the brain barrier, 100%. In our central nervous system, it contacts directly with capillaries, and it can receive information and feedback and actual ions and molecules from the blood through to the brain. Yeah, absolutely. So filtration, absorption, chemical gradients, electrochemical gradients, astrocytes are going to play a role when we're in the central nervous system. Then you have microglial cells. This is a glial cell line, and we can see microglial cells also kind of look like this spider shape. They're a little bit thinner. Their appendages are a little bit thinner. They're also going to directly contact neurons. These cells, you could think about them having these thorny kind of processes. they're going to migrate toward injured neurons, and they can phagocytize microorganisms and debris. So these are going to be your big cleaner uppers. We already had a question about this, about microglial cells, and would they be in a large concentration or a small concentration after cell death or necrosis to certain cell tissue. And they'd be in a larger concentration because they're going to help with cleanup. If there is any dead cells, any infarction or any necrosis or cell death, they can help clean up scar tissue, cell debris, as well as be a big component of our immediate immune response in the brain to any bacterial or outside microorganism. Again, in our central nervous system. We then have our ependymal cells. These are going to range in different sizes. They could be squamous or they could be columnar. They might sometimes be ciliated. If they're ciliated, they're helping with circulation of cerebral spinal fluid, CSF. They're going to line our central cavities of the brain and spinal columns. They're providing that almost that lining to the lumen. We can think about them in that way. And they help form, they actually form our permeable barrier between CSF and cavities and tissue-bathing CNS cells. So these are going to be our cell formers. Think about them lining all the tissues, being that physical barrier between our cerebral spinal fluid in certain cavities and cells. So they're going to help be part of that barrier. They're not doing filtration. They're not communicating with the blood directly like astrocytes, but they're physically lining those structures. I would not talking about any of those specifically. I'm just talking about between, they're going to help take between the cerebral spinal fluid. They're going to be the layer most closest to the cerebral spinal fluid and the structures of the brain or structures of the spinal cord. So the most interior part of any lumen that contains cerebral spinal fluid. So this could be your different draining sinuses of the brain. This could also be directly the spinal cord, the spinal column of the brain. Not necessarily talking about dura, pia, or any of those structures or tissues. Yeah, absolutely. Think of it's your luminal lining. And then you have your oligodendrocytes. Oligodendrocytes, again, are branch cells. Their whole purpose is to process and wrap the central nervous system fibers. They form our insulating myelin sheaths in our thicker nerve fibers. And so you can see here we have axons located here, and we have those oligodendrocytes that are located between the axons. And you can see these are our myelin sheaths with our little gaps. So the oligodendrocytes are going to have processes that form our myelin sheaths around our nerve fibers. So they can help speed up conduction of signaling. And again, this is all in the central nervous system. We're not talking about peripheral nervous system right now. We're talking all about central. There's then two major neuroglia that we see in the peripheral nervous system. It's important to understand the difference between peripheral and central. So we have satellite cells. You could think of them as the astrocytes of the peripheral nervous system. They have a similar structure or similar function. They're going to surround neuron cell bodies in the peripheral nervous system. They're going to help with that structural support and that communication between neurons and blood. You could think of satellite cells in the peripheral nervous system and astrocytes in the central nervous system. You then have Schwann cells. These are going to be similar in function as oligodendrocytes. So Schwann cells are the oligodendrocytes of the peripheral nervous system. They're going to surround all peripheral nerve fibers that have myelin sheaths, and they're going to form myelin sheaths in thicker nerve fibers. This is also, the Schwann cells are vital to regeneration of damaged peripheral nerve fibers. So any nervous system damage, whether it is an actual severing of a nerve or it's maybe compression of a nerve, a peripheral nerve, you're going to see recruitment of Schwann cells to help damage or help repair that damage. So I see a lot of questions where they have the two potential answer responses, or they might be talking about when in a peripheral nerve injury, what type of nerve cell would you expect to be recruited to help repair an injured nerve, something like that. And they might list Schwann cells, oligodendrocytes, satellite cells, and astrocytes. And your answer would be Schwann because it's peripheral nerves, peripheral nervous system, and help with damage. Okay, so then we're going to move into actually neurons. So these were our microglial cells, or our glial cells, are helpers to neurons, right? So now we actually want to talk about neurons, which are the cells that communicate those nerve signals. Neurons at their base are our structural units of our nervous system. So neurons as a whole are these large specialized cells. They conduct impulses. They have extreme longevity. They'll last your whole lifetime. For the most part, they're amitotic, which means that they're not mitotic. They're not going to self-divide for the most part. There are a few exceptions, but that's why they last your whole life, because they're not undergoing typical cell division. They do have a high metabolic rate. So they require a continuous supply of oxygen, which is why, and glucose, which is why when we have a hypoxic state or a hypoglycemic state, our body prioritizes getting glucose and oxygen to the brain because we need our neurons to actually have those components of life. And they're all typically going to have one cell body and one or more processes, which we'll look at here. So the neuron cell body, you often refer to it as the soma. That might be a way that they talk about it in your test. It's your biosynthetic center of your neuron. It's going to be the thing that synthesizes stuff. Proteins, membranes, chemicals. It does have a rough endoplasmic reticulum. It's going to contain a spherical nucleus with a nucleolus. Has some pigments potentially. And in most of neuron cell bodies, it's going to have a plasma membrane that's part of its receptive region that receive input from other neurons. A big part of neurons is they communicate with other cells, right? That's a big component of our neurons. Most neuronal cell bodies, not all, but most are located in the central nervous system. When they're located in the CNS in a cluster, they're called nuclei. When they're located in the peripheral nervous system, they're called ganglia. So if you hear talking about ganglia, that's going to be outside of your central nervous system in your peripheral nervous system. So either somatic or autonomic. So nuclei, neuron cell body cluster in the central nervous system, ganglia, neuron cell body cluster in the peripheral nervous system. This is another favorite test question to throw out there. And often ganglia is the word you're looking for. So what are some of the processes that neurons can have? We talked about the cell body. So some processes that a neuron can have, they have these arm-like projections that extend from the cell body. The CNS can have both neuronal cell bodies as well as their processes. The peripheral nervous system can have neuronal cell bodies, but for the most part, it includes the cell processes more than the cell bodies themselves. But there are ganglia and neurons in the peripheral nervous system. So this is not an all or nothing. It's just if we're talking about where the majority of neuron cell bodies are, they're mostly in the CNS. We know there are some in the peripheral nervous system as well. When you have a bundle of neuron processes, so they're extensions in the central nervous system, it's called a tract. And when you have a bundle of neuron processes in the peripheral nervous system, it's called a nerve. So that's why our cranial nerves are located in the peripheral nervous system. Even though they extend directly from the brain, they're not located in the central nervous system. That's probably one of the most common questions I see people get wrong, because you know that the cranial nerves come from your brain. So you think, oh, they're in the brain, they're central nervous system. No, they are peripheral nerves that come directly from our brain tissue, even though they are literally lying on our brain oftentimes. Two main types of processes we'll look at are dendrites and axons. So dendrites, most neurons can contain hundreds of these. They're short, they're tapering, they're diffusely branched. They are going to contain some of the same organelles as in the cell body. So they do have some metabolic processing that they can do themselves. They're going to have receptive input region of the neuron. So they're the part that's going to receive input of the neuron and then can be able to communicate it back. They convey incoming messages toward the cell body, and they receive those messages as graded potentials, action potentials, which we'll talk about. And these are typically more shorter distance signals. In the brain, you can have even more specialized function of dendrites that collect specific information. And in the brain, the central nervous system, they might even contain little spines or appendages that have bulbous or spiky ends. And again, these are to receive specific chemical messengers or specific messages to certain parts of the brain tissue. But overall, think about dendrites as they have organelles, they have metabolic ability. They're going to have receptive input, and they're going to transmit that up to the neuron body. And that input is going to be as a graded potential. So then our axons. Each neuron has one axon that starts at the cone-shaped area called an axon hillock. Will you have to know that? Probably not. You will need to know that each neuron has an axon. In some neurons, though, axons are short or even absent. In others, they're almost the whole length of the cell, much longer. And some axons can be over 1 meter long, 3+ feet. Longer axons are called nerve fibers. They can have branches themselves. There'll be one axon with multiple branches that can occur. At the end, they're going to branch diffusely, their terminus. And distal endings are called axon terminals. So big differences between dendrites and axons. Cells can have multiple hundreds of dendrites. They typically are going to have one axon. The axon itself can be short or long. The function of axons, they have really efficient internal transport mechanisms, so they can transport molecules and organelles moved along that axon, almost like a highway. Motor proteins, typically in cytoskeletal elements, are what's actually walking them up and down those long axons. Movement can occur in both directions. So you can have anterograde movement, which is movement away from the cell body. So wherever your neuron is, movement away is called anterograde movement. Retrograde is towards that cell body. Often the things that are moving towards are stuff that needs to be broken down, or signaling molecules, viruses or bacterial toxins. They're moving directly towards that cell neuron versus things moving away. Mitochondria, cytoskeletal elements, membrane components, enzymes, things to help these cells do their metabolic functions. Made in the nucleus or nucleolus of that neuron and moved down the axon to the terminus. Yes? It's safe to say that the message goes from the axon to the dendrites to the cell body. The message doesn't have to pass through the axon and the dendrites. So the message can pass through the axon directly to the cell body, but dendrites also contact the cell body. So messages could be moved through the dendrites themselves. But yes, axon terminus, if there's an input at the axon terminus, it's going to move up the axon towards the cell body. It may move through a dendrite, but it doesn't have to. It can go directly from an axon terminus up the axon to the neuron cell body without contacting the dendrites. But the dendrites are extra areas of signal control or signal expression that can also send input and messages to the neuron cell body itself. So both. So think about like one neuron can have dendrites, multiple dendrites coming off directly from the neuron cell body. Then you have your axon. You could have dendrites that are branching off from the axon as well. So it could go axon, could contact some dendrites and cell body, or it could go directly from axon to cell body, or it could go from dendrites to cell body. So it doesn't have to pass through both the axon and the dendrites, a message. The dendrites themselves, their whole purpose is really to move, they can take directly graded potentials, so action potentials themselves, and move them up through dendrites, or they can actually take some molecules or ion input and support, but not as much as axon. Axon's your main transmitter highway for molecules. So if you're thinking about stuff that's being moved, think about your axon highway. If you're thinking about signals or input, axon or dendrites can receive a graded action potential for that input and send it up. Does that kind of help separate? Yeah. When I think of the axon versus dendrites, I think of both of them being able to be able to transmit cell signals, but only one really being able to transmit full molecules or full organelles, and that's going to be your axon. And then we talk about, okay, what is this myelin sheath? It's involved in lots of different pathologies or different conditions. So the myelin sheath is composed of myelin. That makes sense. It's this white protein fat, protein lipid substance. Its function is to insulate the axon to increase our speed of nerve transmission. So this insulation allows for less interference from outside sources. So you have more of a direct send of that signal. Myelin fibers, myelinated fibers are this segment. I have a picture in the next slide. The sheath is going to typically surround most long or larger diameter axons. Not all, but most. Non-myelinated fibers don't contain that sheath, so their impulses is going to be sent slower than a myelinated fiber. So when we see myelination in the peripheral nervous system, that's formed by our Schwann cells. We can see a Schwann cell nucleus here. And then we can see our myelin sheath being almost wrapped around like a curlicue around our axon, which is our axon or highway. And it wraps around like a jelly roll. That's what I think about it as. One cell forms one segment on a myelin sheath in the peripheral nervous system. The plasma membranes then are going to have less protein. You're not going to see any channels or carriers, so they're really good electrical insulators. And you'll see there's almost like these interlocking proteins that bind together, each one sheath, one cell to one sheath, one cell to provide one single layer. But every sheath is made from one cell itself. So there'll be one sheath around, a binding protein, another sheath around, a binding protein, etc. That's our myelination in the peripheral nervous system by Schwann cells. There can be some gaps. And these are sites where kind of axon collaterals can occur. They used to be called nodes of Ranvier. So you might see that on your test still, a node of Ranvier. They still will include that. Now we call them a myelin sheath gap. It's much more descriptive of what it actually is. They are the same thing, interchangeable. They are essentially an opening between the myelin sheath. They can allow for a signal to then be broken off and transmitted in a different direction, or allow for some direct communication from the outside to that axon. And then again, non-myelinated fibers, they're just fibers that don't have myelin around them. They can be surrounded by Schwann cells. So you could still see an axon that occurs that has Schwann cells by it. You have a Schwann cell, you have a Schwann cell, but the Schwann cells aren't wrapping its myelin sheath around the axon. Only when the Schwann cells decide that it's going to be a myelinated component of the axon, they'll wrap around their sheath. But Schwann cells will still exist in the peripheral nervous system around axons, whether they actually myelinate them or not. So the difference then, myelin sheath in the central nervous system, it's going to function a little different than the peripheral nervous system because we have different cells that form it. In the central nervous system, our oligodendrocytes form our myelin sheath, but it's not one whole cell. So like the Schwann cell, it was one cell that wrapped around like a jelly roll. In the central nervous system, you're seeing oligodendrocytes. Each one cell of an oligodendrocyte here can wrap up to 60 axons at once. So it's a lot more of an efficient system, which makes sense. Central nervous system has much less space than the peripheral nervous system, right? We're confined to the brain and the spinal cord. So your cells need to be more efficient. We can't have as many individual cells doing only one function. So one oligodendrocyte can have 60 axons at once or more. 60 is just like a peripheral number of the average, right? But multiple axons that it's wrapping around and myelinating. You can see a myelin sheath gap. So you can see that we have those gaps present here. The thinnest fibers are also still myelinated. And you can see differences between white and gray matter. So gray matter is going to be mostly neuron cell bodies and non-myelinated fibers versus white matter of the brain is going to be regions of the brain and spinal cord that have a dense collection of myelinated fibers. So again, gray matter, neuron cell bodies, non-myelinated fibers, white matter, majority myelinated fibers. It gives it kind of that white appearance. Because myelin is a white lipid protein structure. Yeah. Exactly, yes. and they're in different locations. Oligodendrocytes only in the central nervous system, Schwann cells only in peripheral nervous system. Oligodendrocytes myelinate multiple axons at once. Schwann cells myelinate one axon at a time, and typically you have to have multiple Schwann cells in succession to myelinate a whole axon in its length. Yes. Yeah, so Schwann cells is a great question. So Schwann, we'll go back here. Schwann cells. Where did we go? So Schwann cells, when it comes to their nerve system or their immune system function, Schwann cells are going to help repair damaged nerve fibers, so damaged axons, versus microglial cells are going to clean up excessive cell damage and debris and phagocytize bacteria and viruses directly. So difference would be, I would take a microglia and I would compare it to a monocyte or a macrophage, versus a Schwann cell is just going to directly work on repairing the actual membrane and structure of an axon or a nerve fiber. Yeah. Does that answer your question? Okay, great, perfect. Awesome. So we talked about the general structure of a neuron, but there are specific types of neurons that we do need to keep in mind. So we're going to think about classification of neurons as three main types that are grouped by how many processes they have. So we have multipolar, bipolar, and unipolar, and this is referring to neurons. So multipolar neurons have three or more processes, so one axon and multiple other dendrites. Remember, each neuron typically just has one axon. It could have no axon, right? It could be absent, we're going to see that. But typically, we're going to see one axon per neuron. Again, I told you, it's not 100% system, so you can see here what we're getting to down below. But multipolar neurons have three or more processes, so it's multipolar, one axon, and others are dendrites. This is the most common and the major neuron type in the central nervous system. So if you had to choose one in the CNS, it's going to be multipolar. Bipolar typically have two processes, so one axon and one dendrite. These are more rare. I associate these with more of my sensory or sensations, so sight and sound. So you'll see these in the retina and the olfactory mucosa, but they're more rare. So if you had a question about the retina or olfactory mucosa, and it was asking about types of neurons, then bipolar could be a good choice. Otherwise, you're going to be looking to multipolar for central nervous system, or we have unipolar, also called pseudo-unipolar. This has one T-like process. You can think about a unipolar neuron as kind of occurring like the cell body here, and it has this axon that comes through in both directions. So it's technically could be considered one axon that's crossing through the neuron cell body. So it's technically two axons because it's crossing through in both directions. Our unipolar, these are seen both in the peripheral distal processes and the proximal central processes. So peripherally, distally, we'll see these associated with sensory receptors, right? We're going to have input coming in from over here. It's going to access this neuron and it's going to come out the other side. This is different from having a synapse, right? We have a terminus here and an axon to a nerve cell body, a neuron cell body. Then we have our synapse region. Then we're going to have some other terminus, axon, and neuron cell body. This is different than a unipolar neuron. This is much more common. We typically see this as our communication process when we deal with a synapse, which we'll talk about. This, though, is different. This actually has one central neuron cell body with two axons, or one axon moving in two directions as like a T. So sensory receptors can have this. It's more rare as well. Multipolar is by far the most common, but unipolar can occur probably second most common and bipolar least. If this occurs more proximally, more centrally, it's going to enter the CNS. So distally, it's going to be more likely associated with sensory receptors at the end. Proximally, it's going to be focusing or more likely entering into our central nervous system. So those are your three major types of neurons based off of their structure. We then look at what are the neurons' actual functions or the types of neurons based off of what they do or what inputs they take. So we have our sensory neurons, which are going to transmit impulses from sensory receptors towards our central nervous system. They're almost all unipolar. So again, they're almost all going to have this input, neuron cell body, and output, because they're taking sensation, they're getting some type of signal, and then they're going to send it up to the brain. The cell bodies are located in ganglia. And remember, ganglia is a cluster of neuron cell bodies in the peripheral nervous system. So you'll have some type of terminus here. Sensation happens at that terminus. It's going to travel up the axon. You're going to hit the neuron cell body ganglia, and it's going to travel out along another axon where it's going to synapse with a neuron cell body, typically in the central nervous system. You then have your motor neurons. Motor neurons are going to carry impulses from your CNS, your central nervous system, to its effector organs. These are multipolar. So they're going to have one axon and multiple dendrites. And most cell bodies for motor neurons are located in the central nervous system, except for a few exceptions in our autonomic nervous system. Autonomic neurons. There's a few that are located peripherally. There's then this other type, and this has been a test question I saw. There's interneurons. These are called association neurons. They lie between motor and sensory neurons. They shuttle signals through the central nervous system. Almost all of them are in the CNS. And 99% of our body's neurons are interneurons. Mind blowing. So if you have a question that asks you, what is the most likely type of neuron or most proliferative neuron in the body? Interneuron is going to be your answer. They are the cell shuttlers. They're the middleman of the nervous system, and they're the most common. Are they the most important? Not necessarily, right? They're not necessarily directly taking those signals or directly sending those signals. They are the communicators between the two. Interneurons lying between motor and sensory neurons, shuttling cells through our CNS. So do a little quick memory. The nervous system has three overlapping functions. Which of the following represents a logical sequence of those three functions? Is it sensory, then input, then motor output, then integration? Is it motor output, integration, sensory input? Is it sensory input, integration, motor output? Or is it integration, sensory input, motor output? So which one is it? We have a few answers here. Online, you can post your answers in the chat. I have the chat up, so I'll be able to see. And those of you who said C as the answer are correct. You're going to have some type of sensory input, integration in the brain, and then some type of motor output. That's your typical function, your typical logical order. All right, while studying for exam, you reach for a beverage. To extend your arm, your what nervous system is activated? Your afferent, your autonomic, your sympathetic, or your somatic? Which part of the nervous system is under voluntary control? Would be another way to ask this question. Afferent, autonomic, sympathetic, or somatic? Seeing some right answers. Awesome. Yes, D. Somatic nervous system is your voluntary controlled nervous system that's involved in muscle contraction. So if you reach for a beverage, any action you're controlling in your brain is going to be your somatic nervous system, which is a branch of the central or peripheral nervous system. Peripheral, absolutely. Good. All right, a door slams shut loudly and you flinch. After a few seconds, you realize that your heart is beating very rapidly and forcefully. This is a response as a result of your what nervous system? Afferent, autonomic, central, somatic. Good, great, awesome. We haven't even talked about this. Autonomic, yes, autonomic is your answer. Did it play its little thing? I don't know if it did. There we go, autonomic. That's the same question. Okay, there we go. Complete the following analogy. Electrical wire is to electrical insulating tape, as peripheral neurons are to what? Electrical wire is to electrical insulating tape, as peripheral neurons are to what? Your answer you're looking for is Schwann cells, which are your cells that are helping make that myelin sheath in the peripheral nervous system. If I asked the same thing, but I was talking about the central nervous system, what would we say? Oligodendrocytes, you've got it. Each neuron in our body has a lifespan of what? 120 days, 10 years, 10 months, an average human lifespan. How long do neurons live? Yes, our life, average human lifespan. Why they're not mitotic, right? Which is why once you lose them, you typically lose them. So we want to keep our neuron cell bodies alive as long as we can. Movements of substances towards the cell body of a neuron is termed what type of movement? So movement of substances toward a neuron cell body is termed, is it retrograde, axon-grade, anterograde, dendrograde? Two answers are not actually answers. So you have two that you could eliminate because they aren't real things. Retrograde movement, movement towards the cell body. Anterograde movement, movement away from the cell body. Axon-grade and dendrograde, not things that exist. Portion of the axon that communicates with its target cell is the what? The dendrite, the axon terminal, or the cell body? The portion of an axon that communicates with its target cell. Good. Awesome. Our axon terminal. Yes. Good, good. The basis for differentiation between gray matter and white matter in the CNS is the presence of what in white matter? Unmyelinated fibers, Schwann cells, or ependymal cells. What gives white matter its white matterness? Good. Our myelinated fibers, absolutely. See, this is neuro's easy. You got this. I realize that some of the questions are not the straightforward, but you will get some of these straightforward ones, and you will know them now. What are the most common structural type of neurons in humans? Okay, this is tricky. Oh no, this is not tricky. What are the most common structural type of neurons in humans that are listed on this sheet? Yep, good. Multipolar, most common type of structural neuron in humans. Bipolar, unipolar, pseudo-unipolar is unipolar. Those are the same answer. So since there's two of the same thing, you could have eliminated that one. Bipolar, the least common. Multipolar, the most. If I included interneurons, which would we have changed our answer or kept our answer the same? Changed it to interneurons. Those are the most common type of neurons that exist in the body, but multipolar with these answers given was the correct answer. A few more. I think this is the last one, then we'll take a break. When considering the relationship between a structural classification and a functional classification of neurons, it can be said that all multipolar neurons are interneurons. All motor neurons are unipolar neurons. Essentially, all bipolar neurons are sensory neurons, and unipolar neurons only function as motor neurons. The tricky question, this is more like NFLEX, how they word things to make it not make straightforward sense. Think about what you know is true about these different types of neurons. Think about which is the best answer based off of what you know is true. Trust your gut. C, essentially all bipolar neurons are sensory neurons. We didn't talk about these other neurons being multipolar neurons or interneurons. No, because those are two different types of neurons, right? Multipolar neurons, one axon, multiple dendrocytes, versus interneurons are communicator neurons. So we can't say that every multipolar neuron is a communicator neuron, because multipolar neurons themselves are typically in the CNS and only having an axon moving in one direction. They're not communicating between the two. All motor neurons are unipolar neurons. That would be a misnomer as well. We never even talked about that being a thing. Same with unipolar neurons only function at motor neurons. This is almost saying the same statement in a different way. And so then you're left with all bipolar neurons are sensory. And that is true because our bipolar neurons are only located in the eyes and the nose. So smell and sight. So they're only sensation. They don't have any motor function. So that's your most correct answer. probably closer to how NPLEX likes to word questions because they like to make it confusing. But this one, you could see that B and D were almost saying the same thing, but in reverse. So since you can't have two answers that are correct on the NPLEX, on my exams you can, but on the NPLEX you can't, these ones would go away. And then you're left with saying a statement of all or essentially all. And the NPLEX will typically, I'll answer a question. NPLEX typically an all or a never is probably not going to be your answer. So since this has a qualifier in front of it, essentially all, even if I wasn't sure if that was true, I like that more than the blanket all statement. Unless if I'm 100% positive that it is like a yes, every single time. Most things in the body, there's not an every single time. It's pretty rare when that's a true statement. Yes. For motor neurons, yeah, so we could think about what are our motor neurons? Our motor neurons, let's go back to, here, actually this would be easier if I just end the show and then go back to where the motor neurons were, this card. Oh, no. So most motor neurons are multipolar, but not all. So this is tricky. So you can associate most being multipolar, but not all of them. They're going to carry the impulses from CNS to effectors, and most cell bodies are located into the CNS, except for some autonomic. So if I were going to do something similar to the motor, I would say maybe all motor neurons are multipolar neurons. Essentially, all motor neurons are multipolar neurons, or something to that effect. All motor neurons are interneurons, and all motor neurons are unipolar neurons, something like that. Or you could say all motor neurons are multipolar and located in the peripheral nervous system. All motor, you could say all motor central nervous system neurons are located, or motor, something like that. I think they would probably combine motor and then the type and the location. And they would say something to the effect of all motor neurons are multipolar and located in the CNS. All motor neurons are, or most motor neurons are multipolar, and some are located in the peripheral nervous system. That would be your correct answer. All motor neurons are located in the peripheral nervous system and multipolar, or most motor neurons are multipolar and only located in the CNS. Something where you combine those two things. I think that's what they would do. But most would be most motor neurons are CNS and multipolar, but some could be PNS. And so that's the key with this one. Tricky. So this would be great flashcards to put to memory, separating the types of neurons, the locations and the breakdowns of the nervous system, the types of microglial cells, the specific different appendages that can occur, making sure that you can tell the difference between those on your brain. That will help you with a lot of nervous system questions. We're going to go into some more specific pathways, synapses, and action potentials next. And then we're going to deep dive into the actual specific neurotransmitters and stuff in that autonomic nervous system, because there's a ton of ANS questions on your exam, because naturopaths, we love the autonomic nervous system, and it controls so many parts of our body. So next hour, we'll get into a little bit more generalities of the nervous system, things like synapses, things like all of our neurotransmitters, breaking them down, certain pathways, and then we'll do ANS, and then we'll move on. So we'll take a 5 to 6 minute break. So we'll come back together at 9, let's say 9:17, it's 9:11 right now. And then we'll move into our nervous system part 2 presentation. Those of you online, I'm going to stop my share and I'm going to stop recording.